My name is Levent Aküs. I'm one of the co-founder of CheckImmune and I'm responsible for the project development. CheckImmune was founded in 2019 as a spin-off of the Charité Hospital and it is certified according to 2 ISO norms, and we are working DCLP compliant. Our main offer to clinical partners are our biomarker services, and this from consulting to lab service to medical and scientific data reporting. CheckImmune has a very strong connection to our clinical partners, and with this connection we are able to provide proof of concept studies, which are non-interventional clinical trials, to provide and develop biomarker assays in preparation for the clinical trial later on. The beauty of this connection is that we do have access to patient populations, which is later also targeted in the clinical trial. Therefore, we are very happy to have these connections and to tailor the biomarker program according very precise for the future clinical trial. The ELISA system is perfectly matching to our requirements, because since we are working in clinical trials under regulated environment, we have to rely on very robust techniques with high precision. And with this automated approach ELISA is providing, we can offer highly qualified data for our sponsors and clinical partners.
I'm Andreas. I'm working at CheckImmune, and I'm the responsible scientist for ligand binding assays, multiplexing platforms like ELA, and all approaches sponsors have like these questions for this. The main challenging of validation or developing some assays is the sensitivity and the precision and all the things you need for a whole validation of an assay. ELA is an easy way to validate an assay because it's an automated platform. The standard curve is mainly designed from the publisher or the manufacturer. In context of clinical studies and our validation process, ELA make it much easier. The values of ELA are easy to reproduce, and ELA has a quite sensitivity and short hands-on time, and which minimize errors in measurement, and it makes easy to validate. It has a high precision, maybe this must be it's quite a good system, especially in clinical trials, and sponsors we work together. I think they are fine with this results. The implementation of ELA in our lab was quite an easy way, because the support is really good, and if you have any questions for them, we will come back to you. The ELA you have as an automated platform. You put it in your lab, start the cartridges, have some training, maybe one day, and if you want some certified, you need some Bio-Techne, so on for you. It's really easy, and also the handling too. To train users on ELA, if you don't have a cartridge, you can use the demo mode as the first time to show the technician how does it work, how does the software works, and how you use this device. So it's make it much easier upfront. And in case of the requalification of the technician for using this assay, it's a good tool. And the cartridge himself, you only need some kind of wash buffer. You need your samples, put it on the cartridge, scan this cartridge, and start the assay. Maybe it's one of the easiest way you can measure something. ELA has accelerated our timelines. Can measure 2 or 3 cartridges on one day with all the implementation in our laboratory information management system, with all the protocols we have to write again. Some documentation is always a big big part of our work here in certified laboratories. And for a normal ELISA, maybe we need up to 4 hours, yeah, or 6 hours, maybe depending on the work. And you have the ELA cartridge measured within 90 minutes with pipetting of wash buffer and so on, with starting the device, and you have your full results after 90 minutes to 2 hours. And that's a big advantage, and it accelerates our processes. I think the transferability of an ELA assay is not so difficult like if you have a normal ELISA, for example. Yeah. Because the main advantage is that the ELA is an automated platform, so you have take the protocol from the sponsor, try the protocol at yourself. With your own environment, maybe it has some difference between room temperature and so on. And then you try the assay, but if the assay transfer is complete and works, you have to validate the assay to show the sponsor it works, like in our lab, like in yours. Maybe there's a small kind of validation, like inter-assay is a good way. We had some cases where it doesn't work and we need some adjustments with some normal ELISA. But again, ELA is an automated platform, and I don't know, as I can imagine, that there's any problem to transfer an assay from ELA from a sponsor to our CO. It should be easier than a normal ELISA.
What we see is that there are multiple demands of biomarkers. So sponsors want to understand not even if the compound or if the drug is working, they also want to understand the reason for failure, for example. And therefore the biomarkers, it's a very important topic for the sponsors to watch from different angles and understand their compound in deep and characterize not only proteomics, also transcriptomics with different techniques.
The feedback from sponsors or for ELA measurement is good. We have no questionable feedbacks. Results are reproducible. They are reliable because it's have a good data for our sponsors. We have some public studies. There's a public study done for over 3 to 4 years. The data is consistent for the measurement. There we measure plasma, supernatant, stimulated samples, and the data are really reliable. And we also have some sponsor-driven studies from pharma companies, and the results are perfect. Maybe we have a really good reproducibility of your assays and your measurements.
We rely on good partners to provide good data. So the quality of the reagents we receive and kits we receive, and also the support with our device is highly important. And this is an exceptional collaboration with Bio-Techne, I can say, and also this is in the end appreciated by our sponsors. We are highly rely on that and appreciate the collaboration with Bio-Techne, because the clinical trials in average takes several years, and we have to ensure that the different lots of the kits we are ordering performing in a high quality, and therefore we appreciate the collaboration with Bio-Techne on that, and this is highly important in the context of clinical trial. Yeah, the technical support is an exception, I would say, because whenever we reach out, we get a very fast response, and it's always great to have also a local contact here in Germany, and this helps us a lot in the past years. Yeah, we have to ensure a certain quality, and we also go beyond what is required. So we are doing our own controls and elaborate whether a lot-to-lot change has an effect. We are doing our own bridging of these lots, and was always very happy about the quality we have received from ELA so far.